Yeah. Dr. Kanon, you give a
Yeah,
small
yeah,
one or two line
yeah,
introduction,
yeah,
then I
sure,
will.
sure.
Yeah.
Yeah. So, it's a pleasure to welcome Satendra Kumar, who so readily agreed to give the talk. Let me first congratulate him for getting the Vigyan Yuva-Shakti Award, equivalent to the Shanti
Very good.
Swarup Bhatnagar in 2025, that is this year. So that speaks
Very good.
volumes of, kind of work he has done and being recognized. So we are very proud to have you here with us, Satendra. And what I liked was he is basically a biochemist who did PhD from IARI, and then he kept on. Then he went to Indian Institute of Rice Research in Hyderabad, and all his brilliant piece of work has come out of that. And number two, he also has a variety called KAMALA. I'm sure he's going to talk more about it. How gene editing has helped in creating a particular variety, which is very useful to us. So I won't take too much of time, but welcome Satendra. He is with the ICR Institute in Hyderabad. And without taking too much time, I'll request Professor P.K. Ghosh to say a few words.
Yeah, thank you. I am very glad that we are organizing this webinar, in collaboration with KK Biotech and SBPI. And we are very eagerly waiting to hear about the genome editing genome editing technology in agriculture by Satendra Kumar. So I think that no more introduction is necessary. Let us go to Satendra ji and please start your talk. Thank you.
Sir,
George, John, you want to say anything?
no. I look
Okay,
forward to the talk. I look forward to the talk.
sure. Please start, Satendra. You can show your slides.
Is it visible, sir? My screen?
Yeah, visible,
Yeah.
Yes. You can press on the cup to get the full screen.
Yeah. Oh,
Bottom right.
is it there, sir? Now full screen?
No.
Not yet, because you are not pressed on this particular one. One where there's a cup, the
Up,
larger uh, down in the right hand side.
So then the down right hand side cup is there.
Yes, sir.
Yeah.
It's not actually pressing here.
No,
Wait.
go further on the right hand side.
It's here. I think I need to start. Oh, wait 1 minute, sir. I'll do that.
Doesn't matter actually. I mean, if we can
No.
still.
Maybe I need to share entire screen.
Yeah, entire screen you have to do. Yes.
Is it okay, sir?
Yeah,
No.
hide that. Stop sharing. Next to that, there's a hide.
Yeah.
Yeah.
Now it's okay, sir?
Yeah, it's
Yeah.
lovely, lovely. Yes. Perfect.
Thank you, sir. First of all, let me thank Dr. Kannan and Dr. Charge, Dr. Jain, Dr. P.K. Ghosh, and all the senior faculty present in this webinar. Thank you for sharing this platform. And I am particularly, I would like to thank Dr. Jain for making all the arrangements and also prompting me for this webinar. And in the next few minutes, I will try to cover briefly what we have been doing at Indian Institute of Rice Research, and also what we are doing in collaboration with other labs, and trying to understand the scope of genome editing in Indian agriculture, and as the title suggests, the present status and then what are the major constraints and future promises. So in first part of my presentation, I will cover about very briefly genome engineering and gene editing. And of course, I know every one of you are well aware about these tools and techniques, but then for the benefit of students, I thought there might be some students who will be also attending this webinar. So if we see the genome engineering in plants, it's not that we are doing something new now. It has been happening. Humans have been practicing in genome engineering for thousands of years. And as I have shown here, the variety of rice grains, the carrots, the grapes and banana, all that we see the variation in these food grains or the food commodities. All that variation is coming because of they have the variation in their genome. So the variation has been the key, and that genome we have been engineering, and that engineering was in turn useful for us to create more and more variations that I have shown in this slide here. But now how those variations have been happening, and how did we start the plant breeding. So let us understand, as I said, that the variation that you see in your in the plants around, or in the food items, it is only because of the variation in genome. So the variation in genome, that is what we call genome engineering, has been the key. Now whole of the plant breeding, it has only two the major pillars. One is creating the variations, and the second is selection. So variations and the selections, they are the two major pillars of plant breeding. And it started with the domestication and selection, so the first, if you see the first phase of plant breeding, which has been mostly random genome engineering. When I call it random genome engineering, that means we did the flow of information or the flow of DNA, whatever those populations randomly, and then we selected the right kind of individuals which we might prefer, and then we started cultivating those. So it was to create variation, those random variations, and then select the right kind of individuals. We are revolving around different tools of plant breeding, all that I will be talking about. And it started with domestication and selection. As you know, when we started civilizing, we started living on the banks of rivers. And the first thing that humans, they thought that, how to survive there. And then for this, they started going around and picking the plants with good phenotype. And they preserved those seeds, started growing years after years. But if you see what they were doing, actually they were doing selections. Selections over what? Selections over the variations that was given by nature. So those variations were nothing but the mutations created by nature, humans started exploiting that, going around, picking the right kind of individuals or the plants, and then preserving those seeds. But then, as we started civilizing and the population started growing, and the demand also grew. And we, like other animals, we had brain with us to understand all those processes. Those variations artificially to accelerate the process of getting those variations. And doing two individuals. So when you cross two individuals, you create the variations, and those variations are coming because of the number of recombinants that you create by fusion of two genomes. And those recombinants in the form of different individuals, and then we again the same process. But the only difference was, here those variations were created by us. So we knew that whatever Mother Earth has given us, it was enough to a certain extent, but probably it was not enough. As we started growing, the population started growing, the demand also started growing, and for that we needed to create those variations. And hybridization was one such effort where we started, fusing the genomes and creating the recombinants, and then looking, again, then selecting the right kind of individual. The next we kept thinking, we kept looking for better and better breeding tool, refining those tools, and in this process was the mutation. What mutation could do is to create variations at much faster scale and at much larger scale. So in hybridization, you had limitation of fusion of two genomes, while in mutation breeding, we could take a huge number of seeds and then mutagenizing them either by physical or chemical agents. And then those variations were exploited to select the right kind of, again, the phenotype. So the only difference you see here is how do we create the variations. Selection remain, the critical component of breeding. Now, one thing is very common in all these three things that I have told here, either domestication, hybridization, or mutation breeding, that the variation that we were creating, we did not have control over those variations. Those were random, and then we looked for, the right kind of phenotype. Now, if you look in the second phase of plant breeding, wherein we started creating those variations more precise. So if I want the variation only for a particular example, BT cotton, the variation between the normal cotton and the BT cotton was only, the difference was the the cry gene, cry1. Now, here you could control the variation. You knew the gene that you are going to integrate. You also knew in some cases, not only the gene, but also the locus where you want to integrate. So we could control those variations in transgenic and cisgenic. In India, unfortunately, except for the BT cotton, we could not harness the potential of these tools. And then in this process, the next was genome editing, which actually revolutionized in the past 1 decade in the plant breeding, not only in India, but worldwide. And the reason being is because, some of those apprehensions which people had in transgenic or cisgenic, because of the foreign gene present in the end product, in genome editing, specifically in site-directed nuclease 1 and the site-directed nuclease 2, we did not have any footprint of foreign nucleotides. So they are like completely clean, having not even a single nucleotide from foreign source. And the variation that we created using the genome editing, those were most precise and specific. And that is why it was considered as. Now, when we call precision plant breeding, the precision has been in every area. It's not only in plant breeding that we are moving from random plant breeding to precise plant breeding, but in every field we are now seeing these kind of interventions. We know the precision agriculture, precision irrigation, and a lot of other things. But then, yeah, that's one thing that we have been looking for, and genome editing provides us that opportunity to make those variations highly precise without burdening the plant from any foreign nucleotide. Not going into the detail, but then that is how you create the variation. Basically, it is mutation only in SDN1 and SDN2. You don't provide any template to repair the broken ends of the DNA, and you allow the DNA to repair. In SDN1 case, you don't provide any template. And since there is no template, in some cases it leads to point mutation, or a gene sometimes leading to a knockout of the gene. And these mutations mimic the mutations created by the physical. It is most precise. While in case of SDN2, it is used when you provide also a template strand so that you can do the gene correction. For example, if it was a ATGC there and you wanted to make GATC, so that the negative elements can be corrected there. So what we needed to provide one template, and the template purpose was only to correct the gene, and then it was called SDN2 gene editing techniques. In the SDN3, where you can insert a gene, or you can insert a completely new, you can do exon swapping and so many other things. So SDN3 is it will be very similar to the transgenic. That is why in India SDN3 has not been exempted from strict biosafety regulations. It is only SDN1 and SDN2 genome-edited plants which have been exempted from strict biosafety regulations. Now, when I say exempted, it doesn't mean that it is a blanket exemption. Of course, you need to provide all the data to the regulatory bodies that your plant does not have any foreign nucleotide, and it has got all those. It matches very similar to the plant which was originally it was there in terms of sensitivity towards the antibiotics or the markers that you have used. So all that data, it is there in the SOPs, the standard operating procedure given by Department of Biotechnology, and we need to provide that data. So sometimes people, they confuse that SDN1 and SDN2 are completely exempted. That is not the case. We need to provide the data to show that these plants do not have any foreign nucleotides. The purpose of using here the transgene or the reagent that we send inside the plant cell is to create the mutation. Once the mutation is done, we need to take back everything either through selfing or through crossing by basic genetics, wherein you can segregate these plants which are having mutation but do not have foreign gene. Now in next few minutes, I will cover what in India we are doing with respect to genome editing. First, I will cover the story of KAMALA or DRR Dhan 100, which has been very recently identified in the last AICR-PR meeting. And this has been the timeline. Of course, the planning, dry lab research, tissue culture, standardization. Since it is not the whole presentation in KAMALA, I will cut short. But then it's a very interesting story. Maybe some other time I can speak only on KAMALA. But then it has been a wonderful journey, wherein I would say that 80% of the success depends on how you plan. These are the tools only, gene editing or any other tools come. But then whole of science you need to apply behind that, the gene that you take, the genotype that you select, and not only the gene, that within the gene which nucleotides you should be editing. And this is important to when you know the basics of protein biochemistry, protein structure, you can anticipate the effects and the phenotype when you make those mutations. And it's very important for us to understand that you cannot simply take a variety and a gene, but then there is a lot of science behind. You need to apply, you need to plan all those things. And then developing and evaluating genome-edited lines. From those lines, we created more than 1,000 lines, and then identified the best mutant, which was named as KAMALA. And I will tell why it was best. And we went for deregulation of it. After the deregulation, as I said, we need to submit the data to biosafety regulatory bodies. After its deregulation, then we went for multi-location field evaluation, and then its identification as variety. So when I was working on, when I started working on genome editing, and the first thing was which variety to select and why to select that particular variety. So I selected the variety Samba Mahsuri. The reason being is the Samba Mahsuri is very popular. It is grown in more than 4 million hectare area in country. And we did the SWOT analysis of Samba Mahsuri, understanding its strength, weakness, opportunities, and threats. And the strength of Samba Mahsuri was that it has got excellent plant type. It's a very robust plant type. It has got super quality rice grain that is liked by the farmers and consumers, and it fetches very good price to farmers. It is also very, it has got high demand in domestic and international market. So this was the strength. Then we analyzing its weakness. The weakness was it has got moderate yield. If you see most of the high yielding varieties which have been developed in today's time, they are having 8 to 10 tons of yield per hectare, while Samba Mahsuri's yield is 4 to 5 tons per hectare. So it was important for us to identify the trait. We started analyzing those traits, yield traits, and the yield can be improved by several ways. The first is you can enhance the tillers, but in Samba Mahsuri there was no scope to enhance the number of tillers. The second thing that you can do is enhance the grain weight. But if we enhance the grain weight of Samba Mahsuri, it will lose its USP, which is its grain characteristics that is liked by the farmers and consumers. So we cannot take any chance in terms of changing its grain dimensions, grain quality. So what was left to us was to enhance the number of grains in a panicle. Luckily, we found that Samba Mahsuri has got Sai panicle. It sets hardly 150 to 200 or 220 grains in a panicle, while the high yielding varieties, they go up to 300, 350 grains in a panicle. This is where we thought that we can enhance this, the number of grains in a panicle, and then we selected this particular trait and we started looking for the genes which control number of grains in a panicle. At the same time, we were cautious to select a gene which does not influence the grain quality. So luckily, this particular gene, cytokinin oxidase, it enhances the number of grains in a panicle without disturbing the original grain quality of the cultivar. Already those reports were there. And this was the paper which was published in 2005 by Asikari, a Japanese group. And they were looking for gene which controls the number of grains in a panicle. And the basic strategy to identify a gene or map a gene is to develop a mapping population, map a gene, for which they selected Koshi-Hikari and Habataki. Koshi-Hikari has got nearly 164 grains in a panicle, you can see here, while Habataki has got 306 grains in a panicle. So they developed the biparental mapping population, and they mapped the gene. And after fine mapping, they found that Habataki, which has more grains in a panicle, that is 306, has got mutation in cytokinin oxidase. And that mutation is 16 base pair deletion in 5' UTR and the 6 base pair deletion in exon 1. These two mutations were present in cytokinin oxidase. This particular cytokinin oxidase, which is present in Habataki, while Koshi-Hikari, which sets 164 grains in a panicle, did not have any mutation. Now, what actually at the molecular level it does, Habataki, since it has got mutation in cytokinin oxidase, this mutation leads to compromise activity of cytokinin oxidase. So in Habataki, the cytokinin oxidase is not fully functional. Now, because of this, as the name suggests, cytokinin oxidase, the cytokinin oxidase, it is not able to degrade cytokinin in Habataki to the extent that it is doing in Koshi-Hikari. Therefore, Habataki had more amount of cytokinin in its panicles and in other plant parts. And the cytokinin is the hormone that regulates cell division. Because of more cytokinin in Habataki, due to the mutation in the cytokinin oxidase, there were more number of spikelets in Habataki. If you see in the same paper, although they do not discuss much, but then 5150, it has even more number of grains in a panicle, that is 405 grains in a panicle. This 5150 is a variety present in China. And these alleles have been used in breeding programs in China. And the 5150, which has got 405 grains in a panicle, has got a different mutation. And the mutation is in exon 3. That exon 3 mutation in 5150, it is a 11 base pair deletion. Due to this 11 base pair deletion, it leads to frame shift. And due to this frame shift, there is a complete loss of function of this particular cytokinin oxidase, and it sets 5150 grains per panicle. I just wanted to emphasize that these mutations are naturally present in these varieties. So when we started looking at Samba Mahsuri, we found that it does not have the mutation which is there in 5150, although it has the mutation similar to Habataki, but not to the mutation which gives more number of grains in a panicle, that is the mutation in exon 3. Now we know that we are going to enhance the level of cytokinin, but then it is very important to understand the biology of cytokinin. This is one very interesting commentary published in 2020 in Journal of Experimental Botany, which says that will cytokinins underpin the second green revolution. We know the first green revolution, it came because of gibberellin hormone regulated by SD1 or the semi-dwarf one. Interestingly, if you see the cytokinin, which has got more diverse role in plant biology, has not been exploited to the extent that it should have been. The reason being is the cytokinin is a bit tricky hormone, like we need to enhance its level, but at the same time the enhancement has to be calculated. So for this reason, we created nearly 84 alleles of cytokinin oxidase 2 in Samba Mahsuri by multiplex genome editing. And all that we were doing by taking the clues from the nature, as I said, that is why we targeted exon 3 and exon 4. Exon 3, which is already, mutation is present in Chinese varieties. And these mutations we mimicked to the nature, but at the same time, we did not want to create loss of function of this particular gene. We wanted to enhance the level of cytokinin, but that enhancement should be in moderation. So that is why we had evaluated more than 1,000 lines of those 84 alleles, look for the agronomic performance, and then the best performing line we named KAMALA, and we found that this has got an allele which had mutation, but all those mutations, the deletions are in frame, and it does not abrogate the function of protein. Protein is still there, but it has compromised activity. Because of that, the level of cytokinin has gone up in moderation, and that is why it is giving the best phenotype. And this is in a nutshell. You can see here, if you see the top one is the one that does not have mutation. It is setting nearly, just 200 grains. But if you make these mutations are natural mutations in Habataki 5150 and R498, you can see here. These are natural mutations. And due to these natural mutations, you can see there is an increment in number of grains per panicle. In R498, which is another Chinese variety, it has a combination of mutation in exon 1 and exon 3. And due to this, it does not only have more number of grains in a panicle, but also has got a strong pulp. So what we did, we had taken the clues from all 4,000 rice varieties. We had put all that information together to understand the allelic structure of cytokinin oxidase. And then we did the modeling of the protein to understand that what kind of mutation might create the best allele, which is not, present anywhere. And these mutations we created, which were found in KAMALA, and we not only got the highest number of grains per panicle due to this particular mutation, but also brought two major traits. That is the strong culm, which is there in R498, but also we brought earliness. Earliness is particularly, it is witnessed in southern India, where Samba Mahsuri was actually released. Since the flowering is something which largely dependent on background also, and all these mutations we have done in the background of Samba Mahsuri. Samba Mahsuri is originally adapted for South India. It was released from Bapatla Center. And so that is why we see here in the whole of southern India, it shows earliness. So we got three major traits. And then we have also filed the patent to protect this novel allele of cytokinin oxidase, which not only is showing superior phenotype in KAMALA, but also has got the potential to revolutionize Indian agriculture when it gets diversified in different rice varieties through cross-breeding program. And then we went for deregulation of this particular genotype, that is KAMALA, and this was the first genome-edited plant in India, in any crop, along with the one developed at ICAR New Delhi by Dr. Viswanathan. After getting the exemption from biosafety regulatory bodies, we had entered this particular line, KAMALA, in a All India Coordinated Research Project on Rice, wherein it was tested in multi-location field testing, and mostly its performance was analyzed in the zones or in the locations where Samba Mahsuri is grown or adopted. We found that it shows early maturity. It has got high photosynthetic efficiency. It has got high yield, and also the climate resilience traits, which particularly we have seen at Indian Institute of Rice Research. We have been doing these experiments. It grows very well under less nitrogen and phosphatic fertilizers. Also, it has got a moderate level of drought tolerance. Of course, these are the traits which are analyzed separately, not the part of varietal release proposal. The variety is mainly released for high grain yield. And most importantly, it has retained its original grain quality of Samba Mahsuri. What we see that one particular allele, it is bringing many positive phenotype, and that is why we developed DNA markers for this particular allele, so that it can be diversified in near future in other rice backgrounds, and then it can also enhance the yield of other varieties, not only Samba Mahsuri. And slowly we wish that it gets incorporated in different backgrounds, so that most of those low yielding or poor yielding genotypes can be made high yielding. And here we have shown that if you see the effect that it brings by just incorporating one allele from KAMALA in different backgrounds through marker assisted by cross breeding, it is really exemplary. And then I'll slowly now shift to other efforts in India which are going on, and then here it is abiotic stress tolerance, mainly for drought and salinity tolerance gene, that was DST1 gene. It is well known that the mutation in DST1, it improves drought and salinity tolerance in rice, and for that reason at IARI, the DST gene was mutated in the background of rice variety, another mega rice variety, MTU1010. And they found in their experiment that it gives very high level of drought and salinity tolerance. Although in AICR-PR, this particular variety was only identified as a salinity and alkalinity tolerance, not for the drought. And what we found that this particular variety or the mutant that has been developed under normal condition, it gives yield similar to MTU1010, which is the parent variety, and that is anticipated as well, because the gene, it only helps in is abiotic stress tolerance. So under normal condition, it gives at par yield to MTU1010, but when the plants are exposed to salinity or alkalinity, this particular line, it showed a superior performance over MTU1010, and particularly in the zone 7, again adaptation zone of MTU1010, it showed superior yield of 9.66% under inland salinity, 14% under alkalinity, and also 30% under coastal salinity. These varieties were very recently after they were identified. Of course, there are a lot of other procedures which need to be we need to complete before it goes to farmers. Still the CVRC it has to pass through. Also, such kind of work has been happening in other laboratories. We, in collaboration with the NIPGR, we have made this proof of concept wherein we can make the rice low phosphorus tolerance by editing the promoter element. And here we just made few edits in the promoter, and these lines we have to performed very well under low phosphorus conditions. Of course, this is the proof of concept. We need to, because it was done in Japonica rice variety, we need to attempt similar work in Indica rice variety, which are being cultivated. There are other genome-edited rice lines which are now under multi-location field trials in the background of ASD 16. Here. The second one is ASD 16. This particular line, where the gene edited is AN1 for high grain yield. Again, this is in AVT1. And then MTU1010, this is also in AVT1 developed by ICGEB. And here the mutation was done in DEP1, that is dense and direct panicle gene. All these genes, the mutation is again known that the mutation in these genes contribute to these traits. Those mutations are there in natural gene pool, and it is just creating the similar kind of mutations in these varieties. Then at NIPGR, Dr. Naveen's lab, they have developed this low glucosinolate mustard, and this has already completed, if I understand correctly, the AVT1 field trials of AICR-PR, and it might be now entering to AVT2, that is Advanced Varietal Trial 2. And soon we are hoping that this will be another genome-edited product coming from India. At Nadi Mohali, Dr. Siddharth Tiwari, he has been working on developing vitamin A-rich banana, again using genome editing tools. And sooner or later, we will also see the vitamin A-rich banana coming from his lab. Now we will discuss a little about the constraints in India that we have, and some of you might have been hearing around, the noises. So the first constraint is intellectual property of CRISPR-Cas. Whatever that we have shown here, the tool that we are using, the tool is it was developed by mainly Broad Institute and UC Berkeley. And tool, for example, it is although the variety and everything is indigenous, here the tool that has been used is developed by Broad Institute and UC Berkeley, the University of Vienna. These are the main players. And of course, they have also made some surrogate companies to whom they have authorized these tools, who can license these tools to someone who wants to use genome editing for crop improvement. For agriculture, even Corteva has got the license from these original inventors, and the Corteva can also sub-license to someone who wants to use these tools on their behalf. So one positive thing is that the licensing is not so complex. Already there are examples. Inventors, they are very positive for licensing these CRISPR-Cas tools. In Japan, already GABA-rich tomato, that is gamma aminobutyric acid-rich tomato, which was developed through CRISPR-Cas. It is there in the market. People are consuming, and it is being sold at the same price. So the people's, the apprehension that it will have, all those things are not necessary. And even if because it has got pharmaceutical angle and also the value that because of GABA-rich, so there might be slight variation, but it is not certainly very high priced. The other constraint in India that we are facing is opposition by anti-GM organizations, and you have seen recently a lot of news coverage, and I have put some of those clips here. Now, if we look at opposition of these organizations, here I have put allegations versus facts. They call that gene editing crops are like GM crops. As I have told earlier also that in my presentation, that you don't have even a single nucleotide present in the plant, and that we have to give a proof of it, then only we get the deregulation. So they are not GM, they are simply like mutation product. They have also been telling that farmers will need to pay more amount for the seeds. So big companies will dominate the seed market. Here I would like to emphasize the cost of seed will be the same like any other variety, because there is nothing that, and the burden that farmers are going to bear. And these are not hybrids, so farmers can recycle the seeds. Rather, it is more helpful. The farmers, they will not be dependent on corporates or the seed companies. Here, the case is reverse. Once they get the seed, they can maintain the seed. They are like inbred varieties. They have also been alleging that the ECRIP data was manipulated to release the genome-edited rice varieties, which is not true. All the procedures and protocols were followed with due diligence. Unfortunately, they could not understand the procedure, how it is being followed. And anyway, it's not the part of presentation, but then certainly I can speak on this. The ECRIP is one not only from now, it has been operating since 1965, and more than thousands of varieties have been released through AICRPR. So it's one of the time-tested organization. Both the genome-edited rice varieties, they say, do not perform well. We have seen that they have been evaluated, blind coded evaluation is done, and they have shown superior performance. As I said, the performance is always looked at the target locations where the variety is adopted, not in all the locations. And most importantly, the verdict to accept or reject the varieties, it should be left on the farmers. Every year we release many varieties, and many will fade away. Only good ones will stay. So the technology, if it is there, I think we should leave it to the farmers. They say that genome-edited varieties are threat to biodiversity, but actually it is not the case. As I said, they are exactly similar to the varieties developed through mutation breeding, and mutation breeding has been in place for more than 150 years. More than 4,000 different plant varieties have been developed through mutation breeding. Till now, no threats to biodiversity have been reported in mutation breeding. So there is certainly this apprehension again is not true. Now, if you look back the opposition of anti-GM organizations, immediately it is coming after the varieties have been identified. Although the exemption was given in 2022 for genome-edited plants, and there was wide coverage. I have again put here, the newspaper clips in 2022, when those newspapers, they covered all that news that the government of India exempted SDN1 and SDN2 genome-edited plants. But the significant opposition, it has emerged only after the country successfully developed its own genome-edited rice varieties. Now, fallout of such opposition, let's introspect, critically analyze. It might force unethical research environment because researchers might not declare the product as genome-edited. There is no mechanism by which one can distinguish the genome-edited plant from the mutant plant that developed by other techniques. So there is nothing that one will be able to distinguish. If we have these things in place, people will be coming and reporting everything, and that is what we want to have a very ethical research environment. Most importantly, India will lag behind in one of the most exciting plant breeding and crop improvement field, I would say, because this is one such tool which can help addressing many long-standing issues of agriculture. It can revolutionize agriculture. More than 40 countries, they have already exempted SDN1 and SDN2 genome-edited plants from strict biosafety regulations. Now, the funny part is they have exempted, and as I said, that you cannot distinguish by any means that they are the mutated plant or the genome-edited plant. In future, we will have to import those. We will be importing those plant products from other countries which have exempted, and we will not be able to distinguish also. So there is no mechanism by which you will be able to stop it. Now, genome editing in India, understanding what future promises it has, and the last part of my presentation. There are a lot of things that I can talk, but then I will try to summarize very briefly why it is important. By enhancing the productivity of staple crops that we are doing, for example, rice and wheat, we can strategically reduce the cultivated area under these crops without compromising the total grain yield. That is important to feed more than 1.46 billion people. The land thus saved can be effectively diversified towards pulses and oilseeds. And this will strengthen the production of pulses and oilseed, which is critical for achieving true Atmanirbhar agriculture. Now, these genome editing efforts we can also use to boost the productivity of pulses and oilseeds by creating superior alleles in these crops, by accelerating the genetic gains. And it is also important to understand that genome editing is one such tool that can provide quick solutions to the agriculture problems. Why I am saying this today, it is more relevant when the farmer comes to us. He comes with a problem, and when we start addressing the problem by farmers through breeding, through the existing breeding tools, it takes nearly 8 to 10 years to bring a variety with solutions to the farmer's problem. When we take this solution, by that time the farmer will have another problem. In today's time, it is not important to address the problem, but it is also important to address the problem timely. That is what we have seen in COVID, the vaccines, the timely arrival of vaccines, and the same thing holds true in agriculture. The genome editing can bring the varieties now as early as 4 years, wherein you can have 2 years of validation and testing and 2 years of development. And if we club genome editing with speed breeding, in future we are anticipating that we will be able to bring the varieties as early as 1 year in, 3 years in rice, 1 year for development and 2 years for field testing and evaluation. Now, the sustainable efforts are needed to harness the potential of technology. I am not saying that what has been done is the ultimate, or we have achieved, the best of it. Every technology needs time to evolve and reach its full potential. What truly matters is our commitment to keep working, refining, and improving. With sustained effort and positive mindset, excellence becomes inevitable. I have shown here the cars that we started in 1886, and what now we have seen, just because we did not stop, we kept improving, and now you see the revolution, the improvement, the refinement we see in cars. Of course, we had no choice during the Industrial Revolution. Because of colonization, we do now. What it can do is to create superior alleles of agronomically important genes. All that superior alleles we need to pool through different breeding techniques, through cross-breeding, marker-assisted breeding, or any other kind of breeding tools. Put them together, bring all those superior alleles which have been created by genome editing, and then we will be having the plants which are climate resilient, high yielding, requiring less resources, and all that desired traits, we can tailor them for the local needs. With this, I will conclude. We particularly in India, we are very good in embracing new tools and techniques in every field. In the health, we are okay with robotic surgery. Any new smartphone comes, we will be the first to go and buy from the market. We have shown that encouragement in space science, IT, which has revolutionized our society. Plant breeding also is an ever-evolving process, and it is essential to enrich breeding toolbox for addressing new emerging challenges of agriculture. If we have got richer toolbox with us, we can choose the right kind of tool for the right kind of problem, to address the right kind of problem. It is one of the most precise breeding technology that you can use not only to understand the gene function, most importantly to create superior alleles of genes, developing varieties, and many more applications. What we have developed is 2 genomated rice varieties by ICAR, IARR, and IARI. We have proven that if right kind of environment is given, the support is given that ICAR has given us, we have proven scientific capabilities in India. It also opens a new avenue for addressing pressing issues of Indian agriculture. As I said, it is not the end. It is just a beginning. I am sure if we give right kind of hand holding, right kind of push in positive direction, in the years to come, many more good products, varieties, much better varieties, we are going to get. And there is a whole set of generation that will be working on it to really capitalize its potential. It has sown the seeds and hope for new genome based industry in India. I am pretty sure, like IT, the genome editing is the one which will be one industry, which is going to be an industry in India. If we really support it at this juncture, it is very important. It will not only strengthen, of course, agriculture and farmers, but it will be one of the major job providers to biology and agriculture graduates. So it is just a beginning. Picture abhi baki hai mere dost. We should not say that it is just like why it is not the best. Certainly it is better, but the best will come. With this, I would like to acknowledge everyone who has been associated with me, the director, Dr. Sundaram, Dr. Vishwanathan, Professor Wolf, Vigyan, where I was trained, Dr. Nirja, my head of the department, my team here sitting in front of KAMALA field, and of course the funding support from ICAR. With this, I would like to thank every one of you who are here for patient hearing, and you can see here very clearly the difference growing Samba Mahsuri and KAMALA in field of Indian Institute of Rice Research, Hyderabad. We are growing it in large plot, and more than 5,000 farmers have visited, seen, and also 250, more than 250 officials from industry, from government, they have visited, and they have seen the performance. With this, I would like to thank everyone here. Thank you very much. Yeah.
Excellent presentation.
Shall I answer, sir?
Yeah. You can. Yeah. So the floor is open for discussion.
You see, I think I have heard this presentation and I am actually overwhelmed with joy. Such a nice presentation I have not heard for many years. Actually, this young man has revealed that if the opportunity is given, India can excel and can become a very important player in the production of food grains in the world. Actually, the regulators should support this kind of efforts by properly educating the society who are actually preventing the use of such good technology which are free from the risks of genome conditions. Actually, I had started the regulations of gene edited things and anything of transgenic material in in 1987, a long time back. And we had done very good by forcibly, actually, I should say, introducing BT cotton against all odds. And once I left government, I think after BT cotton, no other technology of transgenic variety has been approved, mostly because the oppositions have been very strong. And that is why we have become a little behind. Now, this boy who has made an excellent presentation and his science is extremely good, I am sure if this kind of people are given encouragement, our agriculture will be revolutionized. Thank you.
Yes, Professor Jain. Yes, sir. Professor Jain. Sir, you are muted. Can you unmute?
I fully agree with Dr. Ghosh that Dr. Satendra has made a very nice and effective presentation. But I would like two observations from him. Firstly, among the various criticism, one of the criticism which really need your observation is that the tool or the technology which we are using is a foreign technology. I mean, what is your comment? Can we think of developing indigenous technology to develop genome edited plants?
Actually, this I have been discussing in different forums. Certainly, if we have our own indigenous tool, we can use it. But then there are few things like when we call indigenous, these varieties are completely indigenous that we have developed. But the tool that we have used it was developed by those two universities that I shared the information. And if we see in today's time, sir, it is time of collaboration and integrating the tools. If we start looking completely from top to bottom, everything indigenous. In fact, I was talking to one of my friend who works in ISRO, that whether do you guys use any foreign tools in when you launch those satellites. So they also use some of the sensors, some of those things which are supported by other countries. Now coming back to developing indigenous tools for genome editing, certainly it is a welcome step. But I think we should not wait for it, sir, because if we wait for these tools to develop and validate, we will be missing the bus. Already, the people, the different countries, they have started bringing products. So we can go parallelly. We can certainly invest on discovering these tools, indigenous tools. But at the same time, we should not hesitate in utilizing what is available for the benefit of the farmers.
Another update I would like to have from you is that sometime back, we heard the news that in the coming years, 15 genome edited pulse varieties are likely to be released. Could you, if you have any information that for which traits those 15 genome edited varieties are likely to be released?
As far as I understand, sir, those are like targets given through this program that Government of India had. And there are different traits. So mostly the traits revolve around enhancing the productivity of pulse crops. I think some of the target genes are very similar to cytokinin oxidase that we have used. So taking the lead from here, they are also targeting cytokinin oxidase in pulses. And along with that, they are also taking number of other targets. But the focus is on enhancing the productivity, and most of those things are the projected targets, as far as my understanding goes.
How about genome editing against biotic stresses, actually?
Yeah, against biotic stresses, it is already proven. It has got excellent scope, and it can certainly help in reducing a lot of pesticides. So since I work on rice, most of my examples I will be taking from rice. Already for bacterial blight resistance, the genome-edited rice varieties have been released in South Africa by Professor Wolf B. Fromer, that I have shown, where I have worked in Germany. And they have edited the sweet genes. And the advantage with the genome editing here to develop the resistance against pathogens is such that most of these mutations we are creating, they are in combination. So it becomes difficult for pathogen to develop resistance, and that is why they are more durable.
Thank you. Thank you so much.
I have
Actually, I am just again coming. We are for the last couple of years trying to decide on the use of modern technology in agriculture in the courtroom. Instead of deciding the fate of the technology in the courtroom, we should try to decide the fate of the technology in the farmer's field.
Yeah.
If farmers feel it is shown that, well, it is working, then only we should decide what country should do. Instead of deciding in the courtroom, I think we have to make a change in our thinking process. So this is, I think, also to be noted, and there is a great need for the important country opinion makers to actually bring in scientifically ways to train the people and to tell them what is good and what is not good. Thank you.
I have a suggestion. I think you know in India the schools, the practicals are disappearing. My personal suggestion is that Satendra, you give the ordinary varieties and your variety. Let them grow it in the lab in their own field. It doesn't need much of an area, and let them see how panicle this thing is happening, how early mature. The children will see. I think somehow the school level, if you promote, they will go to their parents and say, why not we try this variety in their own field? I think that's the only way the mass is going to react. Otherwise, the anti people will keep talking. But I think proof of the pudding is, let us strengthen the practicals in the schools. Like your variety, I think should be given to every school. And the children should do the experiment, and let them experience what they see, let them make their observations. I think I personally feel that will make a big difference than all these debates we do. That's my suggestion, and if I can be of any help, let me know. Because I remember when I was appearing for the National Science Talent Exam, I grew plants, I did this aquaculture, and then tried without various elements how the plant grows. And it was one of the greatest experience I saw as a young boy. That's how I got into research later. I got the science talent, but I think what is a buffer, how to use it, what is alkalinity, how the plant, you talked about, some of the varieties which can go under alkalinity, etc. I think it should all become part of the practical in botany or whatever they are learning, even in class 6, 7, 8. I think we should make it's just a humble suggestion. George, you want to say something?
No, nothing in particular. Nothing in particular, except that dissemination of these viewpoints should be more effectively done. And I would request Satendra to us to upload your slides on our websites along with the entire webinar. I hope it's okay.
Yeah. I think we should do. I'll make the YouTube also, and we'll Pujo want.
Hello.
Yeah, we will provide the link.
And Satendra, I would suggest that even in Hyderabad, and I can help you with some school. You send your seeds, the normal variety and what you have created, and let the children see for themselves. Seeing is believing. It will create a big awareness. I think it will automatically.
Certainly, sir. We will certainly will be doing that, sir. In fact, just that's a very good suggestion. Just I wanted to submit a few points here. That one is that we have been doing this activity as far as the schools in Hyderabad, in and around. We have brought the students, but and we have shown them, and we have seen the level of excitement. They have also seen all that in laboratories. But as you said, we will be certainly, we can do those practicals. The second part is, sir, when we are talking about, the opposition coming, those numbers are very few. If you see what I see every day, sir, nearly 100 or sometimes more than that, either I get messages, emails, calls. Farmers, they are really looking for such varieties. They have seen in the field at IARR. They are really trying for it, for the technologies. So mostly that opposition is not from the large part of the society. It is a very small fraction. Large part of the society, or the large part of particularly those who are the end stakeholders, farmers, they are very, very positive for it. I mean, I can share a number of videos, I can share a number of their emails, messages, calls. It has been so encouraging response by farmers, and I think they are the best judge, sir. So, yeah.
In fact, I feel you must call it technology-enhanced learning. Let children see
Yes.
the technology, how it has been applied, and they start learning from it. I think that is, write a book on technology-enhanced learning or something. It will go a long way. That's my suggestion.
And this reminds me of Dr. Abdul Kalam's quote, in any game, if you see, the players don't shout.
Yeah.
It's only the audience. It is the only audience shout, who are not really playing, who don't know how to play. It's only so kindly please remember this quote of Dr. Abdul Kalam,
Yeah. Anybody else wants to ask a question?
No, I have another one point. Actually, Rakesh ji had already brought it down, but I am again bringing that one. Actually, Satendra had said that intellectual property of Cas9 proteins licensing is not complex. So if this is not complex, then I think we will be able to solve the problem very easily. So can Satendra give a little, a few more sentences to tell how the licensing is not complex?
It's very important first to understand that which claim we are infringing by different patents, by understanding. So every one of us, we are using the tool. So that is very important to understand that which we claim we are infringing. Most of the cases, there might be one or two patent claims. The second thing is they have developed the technology or the tool. Certainly their purpose is that it should go and it should be utilized by many people. So they are also equally enthusiastic to share these tools with right kind of approach that we. So suppose tomorrow I develop any tool or technology, or if I bring something, certainly my purpose would not be to keep it with me only. I would like to have, diversify it as much as possible to give more and more people, so that it will have more and bigger impact. And that is what I have seen. So they are really, really positive. It is very important for us to analyze those patents, understand those claims, which claim we are infringing, and then we can approach. And they have been very considerate in terms of distinguishing the activities which are meant for profit making from the non-profit making.
Okay, good.
So if there are no more questions, may I request Dr. George to give the vote of thanks?
Thank you so much. It's wonderful to have this discussion, and I'm sure the dissemination of your lecture will start taking place from tomorrow onwards.
Yes.
When we put it on the website along with the slides and your entire conversation. So that will be wonderful. And thank you, Dr. Satendra, for agreeing to do this. In fact, I was also kind of chasing Rakesh to help us with one or two lectures and time, but it's worth the wait, and I am sure that we'll be able to do a few more. Thank you so much for joining us.
Yes.
Thank you.
Thank you.
Thanks
you
to
so
all the
much.
participants, Dr. Kannan, who organized the platform. Without the platform, there's nothing that could be done. So thank you, Dr. Kannan, for organizing the platform. Dr. Ghosh for participating. I saw Dr. Bindu, she's also there, and all those persons who have participated in this. Thank you so much.
Thank you so much.
Thank you, sir. Thank you. Good night, sir.
Good night.